Slow start is okay. Yeah, my philosophy is you are graduate students. You know how you manage your time. I yes, so I don't want to force you to be. I mean, not force other people to be. So you have to travel from the medical school to here, right?
Yeah.
Yeah. So
Yeah.
yeah.
But because most of my classes are on Tuesday and
Until
Thursday,
yeah.
I just do all my remote reading and emailing work on Tuesday, Thursday. And
Mm-hmm.
then I live near the medical school. So
Yeah.
I just go to the main campus on Tuesday and Thursday, and then go to lab on Monday, Wednesday, Friday.
Okay. Yeah.
Because there's no point of going back for like an hour.
Yeah.
You know, you
It
can't
is
do
our.
anything.
That's right. Yeah. I think I already put in the slides on the Teams. If you refresh your channel, yes. So this only allow you to put 10 channels, but you have to refresh then force it to see it. So this is week 12 already. So next week will be the last week for the semester for the lecture delivering. I hope you feel it's already covered quite a lot of topics and overwhelmed. So at the beginning, we went to very detailed how the data is manipulated, how the multiplication matrices was done. But afterwards, you'll see, okay, we just talk about the components, the bigger concept. They are just applications, a lot of small algorithm operations added together to show you the applications. So this week we are going to discuss manifold. It's a mathematical abstract concept. And also tensor. Tensor is extension of matrices. Basically, the 3D matrices or higher dimension of matrices. There are some of the analysis if you do at a tensor level, it gives you the holistic analytical power to consider all the different components in the high dimension, rather than just take care of 2D data. So some examples we will take a look. Then at the end, if we still have time, I will illustrate the RNA velocity analysis. And nowadays, people use not sequencing based, but probe based single cell 10x genomics technology like FlexKit. FlexKit, so it's a new, it's hard to see. So 10x genomics had their original product was released 2016 or 17. So at that time, they have a 3 primer sequence-based. But recently, maybe they start to release this probe-based. So this will give you a sequence, but this is like a FlexKit. This is cheaper, and but the data it's generated will not allow you to do the RNA velocity. Okay, it's comparable, so you can. Because it's sequence-based, they allow you to read out the splicing sites. So the gene expression, you have unspliced, spliced information. But for this probe-based, you don't. So no, not comparable with RNA velocity. You just cannot do it. But nowadays, people are using this more and more because they're cheaper. You can put a lot of samples in but you lost information to do this deep analysis. So when you really want to do this area velocity, make sure you know what library you want to use. Okay, so we start by introducing manifold. This is a abstract mathematical object. So you can think about there's an abstract space. This global structure of those data looks very complicated. But there's one simple rule is this object looks globally complex, but on the small scales, if you only look at neighbor, local region, and you can approximate this local region use Euclidean space. Euclidean space means it's a flat, okay? It's a linear combinations of space, of axis. So in this Euclidean space, you have, so if you have A, B, C, make a triangle, then you know AC, the distance between AC is always smaller than distance AB plus distance BC. But this may be not true in the non-flat space plane. If you have a curve, this rule may not hold. So we assume this complex form of the object, which is formed by a lot of data points. But if you only look at the local region, then the relationship between points, you can simplify it into a Euclidean distance space. Okay, so if you think before, we have PCA and MDS. MDS stands for multi-dimensional scaling. So they are similar technique. So those techniques are designed for linear framework. But when you have a nonlinear structure of data and if you only consider the linear projection from high dimensional space to lower, then you lost some of the information between this global information between relationship between data points. So linear algebra, machine learning, data science people start to think we should consider those data points on the high dimensional space. When the relationships are more complicated, we should borrow manifold structure into account. So we consider them. If the data points are close to each other, you can use Euclidean distance to compute their relationship. When they far away from each other, we should not directly compare or study their relationship. We have to consider it's only neighbors, through neighbors, to pass on the information, to learn the relationship between high dimensional structure of data itself. So this is an example. So this S-shaped data points, they are 3D, and they also form a sheet, okay? So we consider this as a manifold. And then if you only look at the data which close to each other, for example, within this radius, in this circle, and then you can compute the distance between your focal point to others. You can compute, say, okay, these 2 points are far away from each other, and this one between this one are more close to each other. So this relationship are true and it's hold. And you can just compute the Euclidean distance to approximate, to estimate. Well, when you consider the long distance relationship between points. So for example, this blue points to any of the red points, you cannot draw a line to use the Euclidean distance as a metric to calculate the distance. What you have to do is to consider the full structure. You have to move from blue to yellow region to orange to red and come back to this. So in that case, the points in the blue region should be far away from the red region because you have to cross the region in the yellow and orange region. You cannot jump directly from here to here. So if we keep this manifold structure, the relationship between points, take into that into account in the algorithm, then we can reduce the dimension from the 3D of this shape into something like this. Okay, so you see this purpose of the manifold learning for dimensionality reduction algorithm is to have a high dimensional data in big D dimension, so they're bigger number. Well, you reduce the points, you map those points to the Y points. Y points belong to a lower dimension, so small d is a small number smaller than big D. In this case, the 3D reduced into 2D. Well, you keep the position of their relative position, and in this case, you will have such kind of dimensionality reduction results. And from here, you can see the blue points have to go through the red, orange, and yellow, and to reach to the red end. Yeah, so this is one of the illustration of this. So once you have this concept on your mind, you can follow this idea and to construct a lot of this so-called manifold learning algorithms. Basically, it's manifold thinking based lineage algebra or machine learning methods. If we only consider dimensionality reduction, then we still have a lot of those options. So I will only talk about isomap and Laplacian eigenmaps and see how they calculate it or internally. So we only open these two algorithms to look at. So overall, the whole idea is you found the local neighbors structures, and you follow these neighbor structures gradually build up. So, so all you need to know is to find the local structure and play your algorithm with small steps. For the long range distance, you have to calculate a series of local steps. Okay, if you look at those algorithms, they all follow this principle. In the algorithm, those steps, they all have the place will allow you to do the local calculation, and their small steps allow you to move along the structure to reach the long range distance. So in the single cell study, we will have transition between one cell type to another, but you have to go through intermediate steps. You cannot jump from one to another immediately. When I see this paper, I was surprised how recent this algorithm has been developed. If you look at the publication date, this December year 2000. So this is the paper introduced ISOMAP, which is a nonlinear dimensionality reduction technique. It's published in Science. So I just have an impression that the whole field is relatively young. Such a simple algorithm with a very simple idea is published so late in the high impact final ejector journal. Okay, so the idea about this is illustrate using figure 3. So this is a very famous artificial data sets, and it's form a Swiss roll. It's called Swiss roll data, which data points form this Swiss roll shape and with the layers overlapped onto each other, but not touched. So for this structure, when you want to compute the distance, for example, from this point to this, and you cannot do the Euclidean distance by their coordinates, they're not correct. You have to follow the whole structure to go from this, the whole thing, and through the Swiss roll and found the parse, which illustrated in the figure B here, panel B. You see this, the shortest parse, which is a real distance between these two data points. So you really need those information when you reduce the dimension from 3D to 2D. So when you expand this, okay, you look at the distance between these 2 points, and this is a reduced dimension in 2D, and that's the distance. And you have to go through the local connections between those networks to reach that level. So once you expand them, and the Euclidean distance and this realistic distance in the red curve are similar to each other. Okay, so the backbone of this algorithm, ISOMAP, is MDS. So we have been talking about MDS from the very beginning of the whole class when we do the dimensionality reduction. Remember the distance matrix, the distance between different cities, you want to reconstruct the map. So that's basically the idea about this multidimensional scaling. So this algorithm starts with a matrix of distance. So if you compute the distance using Euclidean distance on this data, you will get a wrong estimation of the real distance between data points, because without considering manifold structure, you get inaccurate or non-realistic estimation of distance between data points. Okay, so that's why ISOMAP is just found a way to compute more realistic distance to replace Euclidean distance. So we are replacing the straight line Euclidean distance with a so-called geodesic distance. So this geodesic distance is just like in a map. You consider from one city to another. We should not just consider a straight line from one to another, because the region may be isolated with a peninsula, and maybe there's a river, so you have to go around across the street, across the bridge, or the other geographic factors to be taken into account. Similarly here, so you need to consider a neighborhood graph in order to compute the distance more realistic to estimate the geodesic distance. So the whole idea, the whole algorithm is so simple. So distance matrix. So between data points, so they all zeros for the data itself. So this, but the distance is geodesic. And then MDS, then embedding. It's 2D map, low dimension. So that's the whole algorithm. It's very simple. So that's the distance. So how this will, so this again to illustrate. So you cannot just consider Euclidean distance in 3D, and this blue line is not real, so you have to cross the landscape, and then to do the embedding in order to reduce the dimension. And in this case also, you reduce 2D, the data into the 1D, but you have to consider the real distance along this curve to map into the more realistic dimension, distance after reduced dimension. This is one of the example applications to consider geodesic distance. So for the x, you want to compute its similarity. So x is a matrix you get from this is a EEG or functional MRI data from the brain study. So this sample is to compare the similarity between this high dimensional data, which is a matrix, and see whether x is more similar to B or A. Well, in this case, they compute the Euclidean distance, then it looks like it's close to one of them. But if you consider the geodesic analysis, consider other samples as a background, then you have to go from X to B, you have to move through this curve. So eventually, what you found, if you use a simple, straightforward Pearson similarities or correlation as a distance, then this sample X is close to B. But in reality, if you consider the background, follow the manifold structure, then this geodesic distance, which distance between B is greater than from X to A, so you replace you change the direction of similarity comparison. So let's build this geodesic distance when we consider the manifold. Rather than use Euclidean distance from A to B, we consider the structure and then define this distance matrix in order to do the MDS, to reduce the dimension, to give the map. So this map will be the better reflection of the relationship between A and B in the more complex structure manifold. So how can we compute this? So now this is so the algorithm they will use, including to build a KNN. So KNN means nearest neighbor graph. So basically, you build a network or graph. K means you consider how many nearest neighbor as your connected potential connections. If you consider more, then you will have a more dense graph at the end. If you only consider very few neighbors, then you will build a very sparse network or sparse matrix. So this will have some impact on your geodesic distance between same 2 points. Depends on what kind of network you build. Then you will consider the shortest path. Once you have a network, so this will, you have data, okay? Data is very condensed and also in the high complex structure data. Then you build KNN, so KNN will be a network, allow you to connect those neighbors to move from one place to another. Then you have to consider, once you have this, you will start to compute the distance from one travel to another one, and what's the shortest path, okay? There, when you have a complex network, you will have many different possible paths from one point to another. So we need an algorithm called shortest path between any given nodes. So you do this pairwise. You compute all the nodes to all the others and another. So do all the pairwise distance. So the algorithm we can use, including this Dijkstra algorithm. Okay, so this is a very classical algorithm, and there are improved version of this. And afterwards, then you use MDS. Yeah, I like to show the Julia version of those packages, because at that time I thought that Julia will be an open sourced MATLAB look language and for scientific computing. So when people argue, okay, MATLAB is not free, so we don't want to. We always want to use open source free products. I have said, okay, Julia is an open source language and very similar to MATLAB. You can use that. But in general, I don't believe anything open or free is good. MATLAB has its own values. Okay, so the whole idea of this is to model the data using graph-based local affinity. So that's the idea to use KNN, and also, as you said, you can see after you have a KNN, you can also have this minimum span tree on the network to approximate the relationship between points. Either way, you can get the distance, which is a better reflection of the relationship between more complex structure of data. Another way is called using data diffusion. So this is built on the random network random work on the network. So similar idea. So here is to use KNN, then work on the shortest path. Then for the diffusion, we still start from a network. I just use another slightly different network algorithms to do this so-called diffusion process, and to transform this into an affinity matrix to tell you how close the relationship between each nodes from each other and in the network. So this idea, now we just move this, introduce another nonlinear dimensionality reduction technique from the consideration of manifold structure. So this second algorithm after isomap is called Laplacian eigenmap. Which year this published? This published in 2003. So 3 years later, it's the MIT, which group? The Chicago group. So publish another method. So this is based on so eigenfunction of Laplacian Fermi operator. So this is like a new embedding algorithm. So this is the structure of the algorithm. We construct. Remember all this manifold learning, we need to find a way to access the neighborhood relationship. So the best way is to build a network for the data points. Okay, you build a network, then do some transformation, a kernel transformation. Basically, if they're close to each other, then the affinity between them will be high. If they are moving away from each other, you replace this affinity relationship with a certain probability, make them affinity lower. So you use a Gaussian kernel to convert. Basically, the transformation, just replace the weight. Then once you have this network, let's see. Once you have this graph, so this method is to just compute the distance between nodes. This Laplacian eigenmap method is to compute Laplacian matrix from the graph matrix. So this is adjacence, ADJ matrix. Laplacian matrix is a D minus degree minus weight. So you remember, if you revisit your slides, so this formula has been in the spectral clustering. So in the spectral clustering algorithm, we also compute a Laplacian matrix using the degree matrix minus this. Degree matrix, weight matrix, minus this W. W is this adjacency matrix, W here. Okay. So after that, we will do with L. We will do an eigen decomposition for the L. So this is an operation on the this will give you the eigenvalues, which is a vector of lambda. And also eigenvector. So eigenvector. So you have eigenvectors. So you have several probably. So these eigenvectors will be used as an embedding results. So those are the positions of your embedded results. So if you see, okay, I want to use top D eigenvectors, let's D say, okay, I want to use 5 eigenvectors, then those data points from here will use 5 numbers, 1, 2, 3, 4, 5, to replace their coordinates. So you reduce the dimension from the higher into the 5D space, and those are their coordinates. There's some details in this step. When you have a graph, this graph is built by the KNN. You can consider the directed nearest neighbors, so you have all this relationship with them, you linked them. You can also see it's a non-directional, so as long as you're linked, you're linked. It's not direction, no direction. And also you need to, if you want to use a mutual KNN graph, that means this nearest neighbor has to be nearest neighbor to each other's. If you only one direction, then you will not consider this as a mutual nearest neighbor. So you are my nearest neighbor, you have to be nearest neighbor to each other to make a link. Otherwise, you get this more sparse mutual KNN graph. Either way, you get either more sparse or less sparse KNN network, and then you compute Laplacian matrix out of it. Okay, as long as you have a KNN, you will have W, and D is very easy to compute. That's just a degree. Then you get a Laplacian matrix. Then sometimes you want to have introduce the weight. Okay, so now if it's only connected with 1 and not connected with 0, so this will be a very simple weighting schema. But if you want to consider, say, okay, if the distance local greater than 5, then I will trim this into a very small distance, the very small likelihood to be neighbors. So you can have some of this transformation to make some kind of soft cut to make your KNN graph with some parameter. You can change the you can smooth or change some of those connections. So no matter you get weighted or unweighted network, so when you see unweighted, then this Wij is always 1 or 0. If it's weighted, you can have a number which scaled to some level. Then you have weight matrix, which is a network. You have a degree matrix, is add them up for each node. You have a Laplacian matrix, then you do this generalized eigenvalue problems. You will, so that's this step, okay? Then those ND, you select the top from the second, okay? The first one, there's some details. So this first one have no information, so we don't use this, but it start from the second one. From the second, so the first eigenvector will be removed. We consider from the second to the end. Okay. So there are some details in there, because this represents this particular vector represents the overall connections. So somehow they figured out we cannot use the first eigenvalue corresponding eigenvector. So we start from the second largest to move down to the d dimension, depends on how many dimensions you want to keep. So I think we will have chance to practice this on the computers and to get a better understanding of this. Okay. But anyway, so this is just a small example to illustrate. So this example have no real values, but it just show you the code to the operation using a small toy example. So this is for data points example, we build connections, okay? Make neighbors connect to them when they're close to each other. So this network will be represented with the weight matrix W and the dimension degree matrix with this position values in the diagonal position. Node 1 have 3 partners connections, so weight is 3. The other 3 only have weight the degree 1. So the difference between these two matrices become Laplacian matrix. And then we compute this so-called generalized eigenvalue problems. And then to get eigenvalue so this eigenvector will be used as a so this is the largest one, so we don't use this, but we can use any of these two. So this is so-called the second largest eigenvalue corresponding eigenvectors. So for this example, this eigenvectors is 0, -3, and 1, 2. So we can use this as embedding to place these 2D points. The first one is put in the position 0, and the second node put in the position -3, and then 1, 2. So this is a 2D reduced to 1D using the second largest eigenvectors values as their new coordinates. But that's just very wordy description, but that's just the process of the algorithm. Able to achieve this using these lines of code. It's in Julia language, but same in MATLAB. To get the same results. Okay, both PCA and Laplacian eigenmap are dimensionality reduction technique. And the PCA algorithm is linear embedding, not considering the more complex high-dimensional structure. Well, the Laplacian eigenmap is nonlinear embedding. It considers the neighborhood relationship of the data points in the high-dimensional structure, build networks, and approximate their neighborhood relationship, and rebuild, and using rebuild this KNN network, and using network theory Laplacian matrix, this eigen decomposition, to do the embedding. So the process is very different from rotation. It's just directly rotation, no nonlinear change or transformation. as a PCA does. So when you apply this algorithm into this 3-row data set, for example, PCA will give you 2D dimension like this, while the Laplacian eigenmap give you results look like this, and other algorithms, if you check the name, they all manifold learning based, so they can somehow learn the structure. Okay, so those data points are highlighted with different regions, highlighted with different colors, just let you to visualize the structure globally. If you don't have any ideas, the PCA definitely will superimpose a lot of points onto each other, not give you a separable or reasonable reduction view to let you to have some concept about this global structure. Well, the other algorithm, because you can see the transition from different colors smoothly transferred to another, not maxed up. So in that case, the nonlinear dimensionality reduction will do the better job for this case. Some other artificial synthetic data examples, including a shape like this, and this S shape with some holes in the sheets, okay, and this more complex organic shape stuff, and this cylinder with holes in the surface. Then we can see different nonlinear manifold learning dimensionality reduction technique can help you to reduce dimension from 3 to 2 and keep the structure within this very complex shape of the original high-dimensional data. As you can tell now, the power of this algorithm is through study the neighborhood, a small neighborhood network, okay, nearest neighbor network, because that information is in there. So that's why you can learn the relationship between those data points in the high-dimensional space. Nothing magic. Any questions for this part? I think we are moving to applications in single cell. So the most good examples is from this article. This for the fate. We have mentioned the single cell data analysis, the dimensionality reduction technique. We should not use a PCA directly. We can use t-SNE or UMAP if we only care about the clusters isolated. But also this fate is relatively new, and that's the algorithm of fate, which using machine manifold learning to do the dimensionality reduction. So this fate is based on MDS. So it computes the distance between cells. Okay, distance between cells, build a network, and using the kernel to do the transformation to set a soft cutoff, and then you get this KNN network. Then using the information based on the manifold learning algorithm to learn the structure of data. So this algorithm keep the original high-dimensional data structure globally better. Okay, global structure, keeping It's better, I mean, it's better than t-SNE and UMAP. t-SNE, UMAP, you just lost information between this cell type and this cell type. They are isolated islands. You can put those islands anywhere. You can shuffle them. Depends on your random number generator. Well, the fate will keep this subtype and should be globally close to this subtype compared to this subtype. So the relationship is still there. So that's why you will be able to use pseudo time. Okay, you can draw the curve. Did I draw the curve last week? Not yet, maybe. In the MATLAB? I can't remember. No, I didn't do that. So I think I can show you later to build the fate dimensionality reduction. Then you can manually draw the curve rather than rely on the monocle. Monocle, we didn't install that correctly. So so this graph, the KNN graph, the manifold idea is powerful, especially when you consider cell transition are taking small steps. If your sample contains those intermediate stage of the cell, then this will be the powerful approach. And this idea has been always utilized in other algorithms as well. So in the monocle, for example, after dimensionality reduction, we have a so-called DDR tree. This is called a reversed graph embedding. It's actually using manifold as structure information. And the other pseudo-time method, slicer, wishbone, and diffusion pseudo-time, in the middle, they always construct this KNN. So the KNN is for cells. Then using this to compute the distance between cells, and also their diffusion maps or some other transformation of this matrix to approximate this so-called manifold distance. You can see then geodesic entropy, whatever it's called. So all these algorithms has a consideration of the complexity of the data structure. Fate is have a sister so fate is for dimensionality reduction. It has a sister algorithm called magic. So this is just a name, the magic. It's a very similar source code. They basically share the same 80% of the source code are the same. Well, the magic is an imputation tool. Imputation means you consider all this sparse matrix from the single-cell RNA-seq counts, all these zeros as missing values. You will learn the relationship between genes, between cells to fill up those zeros, to replace those zeros with some values. The technique they use is you give a sparse matrix. So you see this is the UMI matrix between cell and the genes with this gene expression counts. And the white space are zeros, so you only have those blue points are the non-zero values. So this algorithm will take the cell-cell KNN graph and smooth them and through a diffusion process. Just you have the structure of network, and you randomly work on the network, see each work, how many times this this parse will go through, for example, cell 1 and 3 all the time, then 1 and 3 should be close to each other probabilistically. If I random walk never pass cell 1 and 2, then cell 1 and 2 should be separated. So by run this random walk, so random walk is just a concept, but when they do the real computation, it's taking, it's not using the real random walk simulation. It's have a approximate with a consider the network as a transition matrix, and then they can do this with a exponential of the network matrices. But anyway, so you can compute the similarity between cells using the diffusion process, and then according to the similarity between cells, you can borrow the expression data from neighbor cells to average out. So you can smooth, even though the cell has zeros in this position for that gene, the cell will borrow neighbor cells, cross, average, and fill up this zero with some values. It could be 0.05 or 0.15, depends on the average of your neighbors. At the end, the output of this imputation algorithm is a full matrix with a lot of those sparse zeros will be filled with some levels of the expression. So this is called imputation. It will be very helpful if you have, say, before the magic imputation, these two genes, because of zeros, you found the cells only have a few of them have a non-zero values for both gene A and B, so the values will be here. Majority of cells have either expression of gene A or gene B are zeros. They will locate it on this X and Y axis. But after imputation, you introduce non-zero values into cell expression. So those cells start to show certain level of expression of gene, either A and B. So you can start to look at this relationship using scatter plot. Okay. So once you have this, all the pairwise gene scatter plot, you can build and start to build a network and make the signal stronger. Okay. So as I said, fate and magic are a pair of the methods which share a lot of similarity under the hood, which they are typical manifold learning, data-driven approach. Another topic is to do the so-called manifold learning. So you consider you have two sets of data. They all share the complexity by its own. Then you consider, I will learn the structure globally from data A, dataset A, and dataset B, and then I will align them according to the similarity in their high-dimensional structure relationship among the data. So this concept was introduced not for single-cell data analysis first, but in the machine learning field. For example, you have this teapot rotate and this 360-degree. And similarly, if you have this, I don't know, it's a toy human body or it's a real, but you also have this rotation, and the algorithm hope to learn this series of picture, the structure of this turning, and you can start to align them together. So this is some concept of the high-dimensional examples of the different data sets, the completely unrelated, which the only thing which they have shared is the way to represent the data in the same way of the relationship between those series picture. You can consider one picture is a high dimensional data in the space, and there's a curve, right, from one and rotate and come back to the same states. Well, on your right side, this toy human, and also each picture is one data points on the space. The shape of those data points also, this circle is back. So the only shared part is their relationship between these two data. So this manifold alignment algorithm will try to learn this structure and make them comparable and put two unrelated separated data sets together, align them in the sum structure. So this is the example. X and Y, the shape are different. The only shared part is from one end to another. Okay, you share this is some kind of rotation. So you want to find an algorithm which you can project this structure, this data, into a shared space and shared with this data Y, okay? Let them to share and map into the same latent space or shared space, so that all those data points can align to each other. Then you can construct some relationship between the right dots in the space in the data X with the right dots in that separate space in the data set Y. So that's the purpose. This is one example. People use the protein structure 3D model. So you have two protein 3D structure in the red curve and the blue curve. Then after you do this manifold alignment, you realize this blue curves, maybe it's a similar protein, just small scaled structure, and then you can align them very well using this algorithm. In the biology, people have been thinking about this to consider the different cells in two different samples. You should be able to align them, okay? So this is a very early stage example. This is called the kernel manifold alignment for the domain adaptation. The idea is to, there are many problems. This multi-view analysis, we need to use the better algorithm rather than just simple CCA. So those are the linear aligners. We will not do a good job, so we need to have some idea about this manifold structure for the data you want to align. So this is one of the. So in this case, this is one early algorithm try to align time series data from the single cell data, and maybe not a single cell data, it's just multiple time points. Try to align those samples with the similar samples collected in the similar time series, but using ataxic, this chromatin accessibility data. So the idea is also using manifold alignment. So those are just examples of manifold alignment used in data biology data. Well, this is called this is algorithm try to align single cell RNA-seq with single cell ATAC-seq. I don't know you know the ATAC-seq, but I will, after the break, I will spend some time to introduce you ATAC-seq. But the idea is you can, but for this algorithm here, is just try to match, okay, try to align them using the NMF algorithm. This is without considering manifold. But I just show you the possibilities. There are other examples. Why it's useful? This manifold learning algorithm will be useful because nowadays we have so-called multi-omics technique. Even for the single cell, you can get single cell ATAC-seq data. You can have a single cell, for example, T cell receptor. You can label the single cell with other protein, surface protein labels. Would that help you to get the same population of cells, or even for the same individual cells, you can have RNA-seq gene expression level and also ATAC-seq, which is open chromatin accessibility level. You get multiple readouts from the same sample. So this techniques have been introduced, so-called single cell ATAC-seq. So you can use this for the single cell in a similar way to do the single cell RNA-seq. We can take a 5 minutes break, and I will come back to introduce this and also the shared multi-modal algorithm. Okay. But I want to go through this before we forget. So the homework, we want to extend the deadline for 1 week because I have to show you how to access or download those data if the data is not in the standard format, because people upload them using wrong format, and sometimes they put junk stuff in there. We need to have examples to show how you handle those situations. And I will send you the link for the course evaluation. It's important for me and for the future students. We have to decide in week 14 what day you want to do the presentation. So week 14 is which date? So this week, so will be 29th, either will be 29, or you want to do it in we can discuss this Thursday and then see you want to do on Tuesday, or you want to do in Thursday in that room, or Tuesday this room. Either way. Yeah. Which one? Final exam date. So this week finished, we can see the final, yes, so will be 6, or you want to do this Thursday, so either way. 5/8. So this will be the final. So this will be the week 15, right? Yeah. So when we have enough people, so we can decide. Either I want to do Thursday for the Tuesday for the presentation and the Tuesday for final, or you want to move to Thursday to give you several days. Yeah. Mm-hmm. Yeah, so deadlines for the homework number 4, homework at 4, deadline move to.
But that was already the thing that was set.
The link. Oh, I should change this. Yeah, I will change it in the Teams. Mm-hmm.
So it's the same deadline that we have already.
It's the same deadline? Okay.
I think so. I think the Teams is
For 24. Okay. Then we just keep that. I saw that I put the deadline this week.
Oh.
Yeah. So you have enough time to do this. Okay. So I confused myself. So the keep the same. Okay. Same deadline. 24. Okay. Yeah.
This homework is what we're going to use for the presentation, or is it?
No, it's separate. Yeah, different. Mm-hmm. You can if you highly relevant to what you are going to discuss. Yeah.
Is it in person or take home?
I will be here. If we decide in Tuesday here, you will just use your laptop, but you can do it at home. Yeah. I will sit here. The only reason is if you have questions, you can ask directly. Mm-hmm. Yeah. Mm-hmm. We have to agree to if everybody agree, presentation and then final, you are done. So we have to vote. I can send a vote, say the most people prefer. Okay. Without a question. So you have 3 minutes for questions. Basically, I give everybody 15 minutes. Yeah. So we have a buffer. You have to come here to plug in those. So everybody have a time limit 15 minutes. With 2 or 3 minutes like questions, and your performance, remember, is very important, because your peers will judge your performance based on the way you present, the way you answer the question, the way you organize things. Okay. Yeah. I don't have a basic structure. It's very just like you do your own research, like your performance report annually, something like that, because you learn the whole semester, and what you learned basically you show us.
So it's more like, is it not on something relevant, like data?
Your own data, you can, yeah, you can use your own data.
Well, I don't have data, so just
Use other published data, build your story. Yeah. Mm-hmm.
But it would be more like, let's say, like a conference presentation, or an
Yeah.
introduction,
Yeah.
the methods,
Yeah.
results and discussion kind of thing.
Yeah. So ataxic, so I will have had I would have had a harder time if I talk with engineering students, but for biology students, it's relatively easy. So your chromosome have heterochromatin and euchromatin structures, and the euchromatin basically they are open and then allow the access for the DNA binding, or there's protein, there are histones, and then this open structure will be allow you to do the methylation, acetylation, those modifications on those histone markers. They all associate with promoter and transcription factor binding. So people want to know the states of the euchromatin structures, whether they are highly open or relatively low closed. So what you can do is to use this sonication to break down fragments, and you can use antibody to fish out the piece of the DNA fragment, and depends on which antibody. So you against this protein, you will learn this particular protein binds to which part of the DNA in the chromosome, in the genome. So when you have this particular antibody binds a lot of those, this particular interested transcription factor, and you will fish out a lot of these fragments, and after you do the sequence, you will sequence more about this particular. You will be able to so this orange one is what we are interested in, and we want to know this particular transcription factor, which region it binds most on the genome. So we use antibody to fish out to bind to this transcription factor, and to get the fragments of DNA out of the solutions, and then sequence them. And then you map back those sequence and see which part of the genome has the peak. So you know, okay, which part of this genome, this particular transcription factor or protein you are interested in are mostly located, the binding sites are located. So those are the basic steps to do this ChIP-seq sequence as well. Then you get this tips of rates if you have a lot of rates mapped to here, so you know the particular protein you are under your study will have a higher chance to bind to this region. So this is a map you can construct. There's some shift in the locations. The algorithm will help you to merge. And then you get a peak of this center of the binding site. Then you build up this map. This map is called the ChIP-seq map. Depends on which protein you are studying. Each row is a particular protein, and you have this particular map, and this indicate this protein have a higher binding affinity in this region with a peak. And then you can look at what are those regions. For example, this major peak is located in one of the UTRs of the gene, and this may be in the beginning of this gene. So you start to have some idea about this protein, this transcription factor and its binding sites and the relationship with this gene under its regulation. So this is the power for ChIP-seq. ChIP-seq is only designed for particular protein. Each time you get one protein's information for their binding site, but this open chromatin structure, which not using the particular protein, so you don't rely on just one protein, or each time you only study one transcription factor, but this is allow you to have the idea how much or how open this region is using this attack-seq technique. So this gave you some global idea about the openness of the chromatin, and you don't know which one, which factors will be bound to here, but you have some idea about this region is more open to be regulated. So this will create, this technique will create a series of peaks, and this peaks is a landscape of your open chromatin structure. So the technique used in attack-seq is particular transposes, and this transposes will cut a bunch to open region of those chromatin. So a couple of years ago, 10x Genomics has introduced this single-cell attack-seq technique. So it allows you to capitalize individual cells and the lysis and do the extract the nucleus, and using attack-seq principle to get amplified tags and then map back. In this case, you will have thousands, tens of thousands nucleus, each come from one individual cell, and then you have this peak information. This peak could be tens of thousands, hundreds of thousands of peak located across the genome. And you can get the information for individual cell, their attack-seq peak, so become a matrix. So this matrix is even larger than single-cell RNA-seq because the number of genes are the fixed, 12,000 or 10,000 genes, but the peak number depends on the cutoff, could be hundreds of thousands. So the matrix is 10 times, 5 times, 10 times bigger than single-cell RNA-seq data. On your lower panel, this is a single-cell ataxic matrix, and the peaks have been sparsely located across the genome, so there are corresponding genes. And you can see those individual cells show very sparse those peak information. But when you take average or combine them, you can reconstruct the single-cell bulk RNA-seq, bulk single-cell, bulk ataxic peaks. So this is example to use 282 cells. When you combine those sparse peak information, you reconstruct the height of this peak. And this is more cells have been combined. So this just visualize the fidelity of the individual cells, so the information can be aggregated. Well, now it create two matrices. One is a single-cell RNA-seq, another is single-cell ataxic. This now, so this is single-cell RNA-seq, as you can have is the cells and the gene 1, gene 2 is somehow transposed because we are using the different format. But side by side, we will have single-cell ataxic. Okay, so I draw this matrix slightly wider than X, so you can see this is Y. Let's transpose it, and also the cell 1, 2, until, and okay. And the peak, now this is a peak 1. Peak 1 can have its location. Its location could be the chromosome 1 and its position in the a number, so 6, 5, 2. Something like that. Maybe I put too many zeros here, but it's a relatively narrow region on the genome, and then the peaks will be the rates located in there. So this, then you have a peak 2, which is another position, your chromosome 1, and then 2, so small region, and 1. So this will be a very, very big matrix, contains many, many peaks. So most recently, 10x genomics allow you to do this ataxic. So this is ataxic, single cell, RNA-seq. So it allows you to do this for the same cell, and then the cell 1 here is the same as cell 1 here. So in that case, makes the manifold alignment easier. So you don't have to align them because you know they're aligned. Okay. But for the more generalized technique, you don't have that information. They're completely matched. So you get the population of cell from 1 to n. In the here, you may have different cells, all from the same population, but there could be the different cells. This could be different sets compared to this. In that case, if you believe there are some of those cell structure here, so based on the profile, these cells form shape like this, some cells here, some cells here, and then you will expect from the same population use a different metric, either ataxic or other protein concentration. You will have a structure similar to this, okay? The cells also depends on your profile for this model of the data, and you have this shape. You want to use manifold alignment to align these cells. Why you want to align them? Because you want to know a cell which here, and its corresponding cell should be somewhere here as well. So you know, okay, this particular cell, orange, aligned with this cell here internally. And then you can learn those genes for this particular cell. This gene expression may relate it to those peaks to make this alignment to help you to interpret. So those called multimodal data. Yeah, nowadays we enjoy this single cell RNA, single cell ataxic from the same nucleus. Then we don't have to do this alignment. But that's for the future. If you have something like this in the multi-omics integration age, you have a single cell population, then you can have RNA-seq, chromatin ataxic. You may have a protein concentration measured from the same population of cells. You also methylation. You have DNA, genome DNA. You don't expect for just real individual cell, you can have those multiple omics at the same time. You shouldn't have expected so much. In that case, we will get multi-omics data, but we need integration. So this integration will rely on the manifold alignment between two sets of data measured from a same population of cell, but different individuals. Nowadays, if you have single cell ataxic, you can use the extension of Surette package, the single neck, okay? Single neck is R-based to help you to do these similar things, okay? To do this basically is a Surette version for deal with single cell ataxic data. So there are a lot of similarity, but there are slightly different ways to treat the sparsity in here and deal with the peaks rather than genes. I don't know what current the Surette version is, but the version 3 contains some of the information about how you do the integration if you have a multi omics data from different technique. Okay, so this is just about the manifold alignment. So we start from dimensionality reduction, the concept of manifold, move to alignment. Now we talk about the different topics, which is tensor, as I mentioned. Tensor, you can think about this. If you have a tensor of rank 3, basically the 3D array, 3D matrix. It gave you higher mode structure and higher dimensionality. But normally we don't work on the tensor because it's computationally expensive. It's very hard to handle. We work on the numbers, vectors, matrices. But you'd better to know there are tensor-related algorithms existing, and we do have in the real situation many data can be represented in the tensor form. So this is a 1D vector, 2D matrix, and a 3D or third order tensor. You can have a higher tensor structure in your imagination, the matrix, okay? So real tensor example data. So the color image, if you know there are 3 layers, if you look at the image files, each layer the density of that particular channel represent this blue, green, red. Okay, you have 3 matrices superimposed onto each other. So this depends on the dimension of your image. This could be 300 by 400, but you have 3 matrices. So the total, the data is a third order tensor with a dimension 300 by 400 by 3. Okay. And if you have, this is a static image, that if you have a video, then that video will be a time series of image. So it increases one more dimension of time. So now you have a 4th order tensor, which color image by time series. So this is an example for the tensor structure. And you can put, for example, this 2D images. Okay, many of them you can put them together, make a tensor. If you have this image file, you make an array of this by similarity. Or there are many other high-dimensional tensor structure data. In physics, and all these parameters are tensors. Okay, it contains information about location and also other dimension information, maybe time. So it used widely in the physics world. So some languages you can functions writing in the language, if you consider take tensors into account, then you have to code independently, differently. If you think your input data is always matrix, then you don't have to consider the 3rd dimension. Okay, but if the program you are writing, the function, wants to take tensor as input, you have to deal with this slightly different. If you look at this code here, the data contains coordinates x, y, and the layer z. So this is like 3D information. And in the computer science and mathematics, people sometimes use different terms, but they are interchangeably, and you can see the number or scalar or array or vector, 2D array in computer science, and in mathematics people say matrix. For the higher dimension, the ND array, and here you can see ND tensor. Well, remember we have done the matrix decomposition. We want to learn the major components within a matrix, which the product of those major components can be used to reconstruct original data matrix. We can apply the same idea for tensor, and to do the decomposition depends on how what's the order, how many dimensions in the tensor. You can decompose them into multi-dimensional vectors or the product between matrices. For example, in the I should have that. So this is matrix decomposition, and remember this is SVD. Okay, you decompose A into U and sigma V transpose, 3 matrices, and you can use major components, only 2 to construct the whole, approximate the original matrix. Okay, if you forgot this, you have to revisit the composition, the lecture. So this is a way you can extend this idea into a tensor. Now, the 3D, this qubit, will be decomposed into one matrix, another matrix, and a third matrix, instead of 2, and we have 3, and in the middle there's another smaller qubit of the data, and plus the whole noise. And then, so if you ignore this whole thing, which contains the noise components individually, you can use this 3 matrices plus this to do the multiplication and reconstruct the whole thing. So we are interested in the values in those components because they learned from multiple angles to see how we can reconstruct to fill in all the values in this qubit instead of this matrix. So those values will be very useful to tell you how this multi-dimensional matrices or tensor was generated. So that's the idea. Turn out in the history of the tensor decomposition, there are 2 lines of researchers. They take a different approach to do this decomposition. One is to simulate this similarly as a low rank approximation. So remember, we can approximate X with M, and M can be reconstructed with a outer product between vectors. Okay, A1, A2, A depends on how many major components you want to keep, and each one will generate a matrix, and just like multi-layers matrix, and their summation will be the M, and M will be very similar, or will be the approximate of X. So this is a low rank decomposition. And so the one way of the tensor decomposition is borrow this idea to decompose a high-dimensional tensor into multiple vectors multiplication. So what we can do, we can see, okay, this is a component 1. So the first components will be the multiplication between vector A1 times vector B1. So this will form a matrix. And then you will multiply it with the C1. So this will create a 3D qubit of the data. Okay? And then you have another second components, A2, B2, C2. and the multiplication between these three vectors give you another qubit of the data, which is the size is the same as m. And then you put them together by add elements to each other. So each element to each element, you can reconstruct the m, which is a low rank model of the original data. So if you keep the top components, you will be able to approximate x and reduce the noise in x. I hope this makes sense to you. So this low rank structure, this is called the CP tensor decomposition, or CP tensor factorization. There's two different naming systems. They all called the CP. Okay, the one is called is from this paper, and then the other from this paper. But anyways, they are the same. So how this be? How can you learn? Basically, you want to learn from the input tensor, and how can you learn the values in vector A1, vector B1, vector C1, A2, B2, C2. So this is this requires the algorithm to learn these values. Okay, so this becomes the optimization problem. The algorithm will do a loop, okay, iteration. It will start to approximate one of the dimensions. So we see a dimension. So, and then do the next, and then do the third. So you just take a turn, minimize 3 steps, and then come back, loop again 3 steps. Just do. Then at the end, you achieve this so-called this procedure is called alternating least squared, and then at the end you achieve the least squared approximation, right? There are techniques which can accelerate this by random sampling, basically, to reduce the computation requirement. So this is called a CP low rank approximation for the tensor. Another fashion, another way to do the decomposition is called the Turk low rank. So this will decompose this tensor, like 3D tensor as an example, into several fibers. Okay, so those fibers will be used. You can reconstruct the whole tensor again. So this represents a very different ways to do the decomposition, but there's only 2 major decomposition methods. Let's see some applications of the tensor decomposition. So this is a sparse binary data sets from the study of the social network, sampled from a group of students, and they have time series data. So every hour probably they sampled the whole network. So at the end, you create this time evolving social network connections, and then this is automatically become a tensor. You can learn from the tensor decomposition and what's the frequency or the peak time of sender and the receiver, and there are many other decomponent fractions. The components they can learn from, and to learn from this data to get some idea about the patterns of this social network evolving. Another application, so you can get those explanation of those tensor learning or tensor decomposition applications through this YouTube. The speakers discuss those applications very nicely. This is the applications in the in the neuron and the behavior study. So they have the probes in the mouse brain and then look at the monitor the neuron activities across the time, and also their information about where and what this mouse is doing, and they are doing the right turn or left turn in this cross structure channel. They also have the award, say, okay, this mouse traveled to the west end and through the turn from north and get an award. So they collect this information and also the multiple trials and over multi-days and multiple samples. So they put everything together, the huge data set to learn the tensor major components. They can find the activities of neurons which belong to which time framework and what they are doing. They found some neuron activities associated with the right turn of the behavior, and the sum is after the giving the award. So if you don't have those learned components, you don't have those values, you will be just a huge set of data which contains messy signals of the neural activities. So in the talk, I believe the speaker, she described those separation of those neural activities with behavior changes. So each component tell you a story. So this is an application in my own study. So in this case, is 24net. So we have a sample, 2 samples, and we have subsamples of cells. Each subsample we create a gene-gene interaction network. So gene regulatory network for each sample, and then this become a tensor. So you have multiple networks, same shape, but depends on your subsamples. We have T networks. And then because there are 2 samples, you have a black and red. So this makes another layer. So this like a N by N, so N genes by N genes network, and T samples, and also 2 categories. So make a four-dimensional tensor. Then we do this decomposition and then learn the major components, and then reconstruct and learn the major components, reconstruct tensor, take average. At the end, we can get a very smooth and denoised version of this network, because each individual network may be very noise, but take average, you learn the major tensor, and reconstruct the ones really, really clean. So that will help us to do the alignment of network to find the change of the network nodes between these 2 samples. I think that's all for today. We will not do that RNA velocity again by giving the time. And so this is today what we covered, and then next week we will talk about the networks, okay, for the gene networks, the cell networks. Okay. Any other questions? All right, see you Thursday.